Hello, my hardworking students. So in this video, we will focus on a variety of chemicals, common chemicals that are typically released in the tissues during the process of inflammation. That is, of course, injured or damaged tissues. So just a quick reminder of where we left off last time. We discussed the vascular and the cellular phases of acute inflammation. And so here we're going to focus on just this subtopic, the chemical mediators. In acute inflammation, there are three major groups of what we call chemical mediators. There are these amine-based chemicals, lipid-based chemicals, and then we'll group together the inflammasomes and the cytokines. Vasoactive amines are amine-based chemicals that are going to change how leaky capillaries are, and they're going to change the diameter of blood vessels. Many of them are going to cause vasodilation, but some of them may cause vasoconstriction. For instance, you've heard of histamine. We all have heard of histamine because many of us take antihistamines. Clearly, that means that we're taking medication to fight off the effects of histamine. And if histamine is a really, really good vasodilator, makes your capillaries really leaky, and which is why your nose is running, which is why your face looks like it's as big as a balloon when you have bad allergies. Another set of chemical mediators are called the eicosanoids, and they are lipid-based. They're also, because they're lipid-based, they can easily slip into cells through the phospholipid bilayer. So they're very important in cell signaling as well, in all cells, even the platelets. So again, these eicosanoids, they promote inflammation. They may even promote fever, allergies, and we'll take a look at some very specific examples here. So before we take a look at how these eicosanoids actually impact us in inflammation, I'm showing you here a table with what I think you should know as being the three major examples of eicosanoids. So they're all going to function in some aspect of the vascular or the cellular phase of acute inflammation. Notice that the prostaglandins also promote fever. As a matter of fact, the prostaglandins, and a very common example of a prostaglandin PGE for prostaglandins PGE2, because it actually communicates with the hypothalamus, more specifically the temperature center in the hypothalamus, such that when prostaglandins are released during inflammation, they can cause a fever. When we have an invasion of a pathogen, we often end up with a fever. So these are your three main eicosanoids, your prostaglandins, the thromboxane A2 one, and then the leukotrienes. So where do we get these eicosanoids from? Believe it or not, we get them from the phospholipid bilayer in our cell membrane. So we're going to go over this text slide briefly, and then we'll look at an actual diagram to better understand it all. But let me get you set up a little bit here. So there's something called arachidonic acid, a very important chemical in our body that is a fatty acid, I should say, and we obtain it from our phospholipids in our cell membrane. So the way we manage to retrieve it from those phospholipids is by using an enzyme called phospholipase. Now, in order for phospholipase to do its job, in other words, in order for phospholipase to be active and essentially split up these phospholipids so that they can free up of the fatty acid called arachidonic acid, they need calcium. Now, you also know that we've learned in the slides on cell injury that cells can actually go into calcium overload. And so now you can put two and two together. If there is way too much calcium, all of these phospholipids are going to be broken down to give up their arachidonic acid so that they can be converted into eicosanoids. And what does that result in? Lots of inflammation, obviously. Now, arachidonic acid has many functions. Again, because it's a fatty acid, it can very easily slip through the phospholipid bilayer and function as a second messenger, or basically a signaling molecule. It can regulate other signaling molecules, signaling enzymes, and it can itself cause vasodilation as well. So we'll take a look at a diagram now to put this all in a better perspective. So let's focus on this this diagram with all these different biochemical pathways. It looks a little overwhelming, but we'll figure it all out. Don't worry yet about all of the text and these extra arrows I created. So here we have our phospholipids in the cell membrane. They can be broken down with the help of phospholipases. Remember, they need calcium in order to become activated, and then that releases the fatty acid called arachidonic acid. Arachidonic acid can then go through various conversions with the help of enzymes to then become some kind of an eicosanoid. So here we have a prostaglandin that can then become this kind of a prostaglandin. Here's your PGE2. Remember, that's your fever-causing prostaglandin. This prostaglandin can further be manipulated or converted into prostacyclin, another type of prostaglandin that you'll hear a lot about, or thromboxane A2. On this side, you see a variety of leukotrienes listed, right? So your three major eicosanoids I asked you to remember are the prostaglandins, with a good example, PGE2. There's also thromboxane A2, and then the various leukotrienes. Okay, now I'd like for you to take a look at the following. Once again, phospholipases are needed to literally release the arachidonic fatty acid from the phospholipids in the cell membrane, or even this diacylglycerol. Don't worry about that so much. I'd like for you to know about the phospholipids. And then we have these COX enzymes. In particular, I want you to notice that are needed to convert the arachidonic acid in our prostaglandins, including thromboxane. So what is one of the first things you go do when you don't feel so well? Well, you go buy some ibuprofen, or you take an aspirin. You take those non-steroidal anti-inflammatory drugs. Well, these NSAIDs, or singular would be without the S, and non-steroidal anti-inflammatory drug, they are going to prevent the working of these COX-1 and COX-2 enzymes. And what do they do therefore? Well, they prevent the conversion of arachidonic acid into these eicosanoids. These eicosanoids, as you know, promote inflammation. So if we prevent these enzymes from allowing this conversion, we can reduce inflammation and hopefully feel better. At times, people are prescribed corticosteroids to reduce inflammation, and they act at an earlier time. They actually block the phospholipases. So here you see how, very complex bunch of biochemical pathways are not looking so overwhelming anymore. They make a whole lot more sense, and you can now better understand why we take some of the drugs that we take. Another set of chemical mediators that are produced during acute inflammation are called inflmaasomes, and they're proteins. They're good-sized proteins, and they're going to increase the production of cytokines that like to create inflammation. So they're going to be they're going to be chemicals that stimulate the production of even more cytokines. And we'll get to cytokines next after this slide. They're produced by a whole bunch of immune cells. I'm not even going to ask you to memorize which ones specifically because they're produced by so many. They're particularly activated by the TLRs or the PRRs that are binding to the PAMPs and DAMPs of damaged cells or pathogens. So we get to our final chemical mediator. We've been talking a lot about cytokines. We've been mentioning that they're secreted by all of these immune cells and that they play a very important role in telling what for instance, the residential cells might be secreting them or the macrophages might be secreting them, and they inform other white blood cells in what to do. So they're tiny proteins. They're not like the inflammasomes, which are bigger proteins. These are tiny and play a very, very important role in cell signaling. Notice that cytokines are not only produced by immune cells, but really by all cells in the body, except for the ones that do not have a nucleus. And that means, not red blood cells. Red blood cells are not nucleated. You all know that by now. They are proteins, and because they are proteins, they cannot cross the cell membrane. Only lipid-like molecules can easily slip through the cell membrane. So the only way they can trigger a response as a cell signaler is by binding to the cell membrane. Remember that I used the abbreviation CM for cell membrane. Some people refer to them as hormones, but because many hormones are typically chemicals that signal to cells, we shouldn't do that because these guys occur in a much, much lower concentration, unless there truly is cell injury or an infection. Then they're very rapidly increase. So the production of cytokines can increase very, very rapidly. So we're going to look at a few important ones. The interferons, there are the interleukins, and then there are the tumor necrosis factors. And we've mentioned the chemokines already, which are chemicals that attract other cells by means of positive chemotaxis. As I said, we should not include the hormones and also not growth factor. You might have heard of what would be a good growth factor, insulin-like growth factor. Those are just too big of a molecules to include into our group of cytokines. So let's start with our first set of cytokines called the interferons. And as the name says, these interferons interfere with something. They interfere with various things, mechanisms in particular, to where it becomes much more difficult for a virus to survive, let's say, or for cancer cells to survive. So let's say a cell has been infected with a virus, it's going to crank out or crank up the release of interferon, and the interferon molecules are going to start binding to neighboring cells. And those neighboring cells are now going to go through these biochemical pathways that are going to make some changes to the inside of those neighboring cells. For instance, their protein synthesis mechanisms might be altered somewhat to prevent it, to make it much harder for a virus to survive inside of them. Now, you haven't learned much about invasion of viruses in this class. You might have, at least in microbiology, but viruses need to make proteins to create more of themselves. So if they can't do that, it's going to make it very difficult for them to create more virus particles or virions. Not only that, interferon can start cranking up the activity of the protein p53. Remember, that is your tumor suppressor protein, which has the responsibility of stopping the cell cycle to make sure that the DNA that's damaged has a chance to repair itself. Well, let's say that a virus has begun to really damage the DNA of a host cell, or a cancer cell has DNA that's very mutated. The p53 um, protein, as you know, can set off a series of reactions that then activates the caspase enzymes to where we see apoptosis occurring of that virus infected cell or cancer cell. The interferons can activate natural killer cells and macrophages and more, as we'll see on the next slide. They can also increase the defense mechanism of a host cell by upregulating the amount of antigen. They can also prevent the growth of blood vessels by tumor cells. Tumor cells, in order for them to be able to grow and divide, need nutrients, which means that they're going to secrete all kinds of factors to stimulate the growth of blood vessels around them. We call that process angiogenesis. Genesis, the creation of, angio referring to blood vessels. Um, so these interferons can interfere with that particular mechanism that cancer cells like to depend on. They're also the source of fever and give you those flu-like symptoms, by the way. Another set of very, very important cytokines are the interleukins. As a matter of fact, they're so important that our immune system literally could not function without them. And when there are problems with interleukins, one or more, we are indeed going to see things like autoimmune diseases, which we will study, or immunodeficiency diseases, which we will also study. Interleukins are mostly produced by your T helper cells, although all kinds of cells can produce them. And they're going to be very important in the red bone marrow and the process of hematopoiesis. So let's take a look at a picture to prove this. So this diagram shows you, of course, the whole process of hematopoiesis. Here we have our hematopoietic stem cells, sometimes called the hemocytoblast. It produces its precursor stem cells, and then we get our platelets, our various white blood cells, and here we have our erythrocytes, right? We're not really that interested right now in all the different cell types, but what I wanted to show you is the number of cytokines that are needed in order to create functioning white blood cells, platelets, red blood cells, etc. All the ones that have CSF in them are some kind of colony stimulating factors. And of course, then the interleukins are IL. The final cytokine that we'll focus on is the tumor necrosis factor, or TNF, which is often referred to as cachesin, a little difficult to pronounce at times. And it plays a very important role in regulating our immune cells. Again, it's a cell signaler, and it can cause the things we have been discussing: inflammation, fever, apoptosis, but also cachexia, which is, we'll talk more about this when we learn about AIDS. But when I mention AIDS, typically I think many of us, especially if you're more from the generation that was familiar with the 1980s when AIDS was rampant and not very well understood, many people became extremely emaciated towards the end of their lives, and that is a form of cachexia. So they might have the proper nutrient intake, but they just cannot put any more weight on. And this has a lot to do with this new tumor necrosis factor. This tumor necrosis factor can also prevent the creation of tumors, tumorigenesis, meaning the formation or the creation of tumors, and make it very difficult for viruses to replicate. So that wraps up our discussion of chemical mediators. Quick reflection. So we studied the vasoactive amines. Remember, histamine is a really great example of that. Then we discussed the eicosanoids, and we talked a little bit about how our over-the-counter aspirin and ibuprofen can prevent inflammation by preventing the formation of eicosanoids in the arachidonic acid pathway, right? Or even the corticosteroids can do that by blocking phospholipases. We briefly mentioned the bigger proteins called inflammasomes, but we spent quite a bit of time discussing the cytokines, which include your interferons, which include your interleukins, and also your tumor necrosis factor, as well as your chemokines, which are the kinds of cytokines that attract more white blood cells to a site of injury by means of positive chemotaxis. So, are we anywhere close to finishing up our discussion of acute inflammation? I hate to tell you, not quite. So, in our next presentation, I should say in our next video, we're going to focus on the plasma protein systems primarily, and the hormones is just a small section. And then there will be one more video on types and outcomes, and that will then wrap up the discussion on acute inflammation. After that, we still have to discuss chronic inflammation and the systemic response towards acute inflammation as well.